Very good morning to all of you. So today's our topic of discussion is that of apoptosis. Okay, apoptosis. Apoptosis is very important topic for your short note UG and PG both, and especially for PG, it's very important short note. Okay. So apoptosis. What is apoptosis? Apoptosis is nothing but it is a programmed cell death. Okay. What is apoptosis? Apoptosis is a programmed cell death. Okay. So how now comes on the definition. If definition will come, then how you will define the apoptosis. So how you will define apoptosis? Apoptosis is a, what is apoptosis? You will define apoptosis. Apoptosis is a pathway. Apoptosis is a pathway of cell death. Apoptosis is a pathway of cell death in which what happened? In which cells own enzyme, in which cells own enzymes degraded its own nuclear DNA, nuclear and cytoplasmic proteins, Okay, so this is the definition of your apoptosis. So what is apoptosis? Apoptosis is a programmed cell death, and actually apoptosis is derived from a Greek word. Okay, it's derived from a Greek word, and in Greek the meaning of apoptosis is drooping off. In Greek, the meaning of apoptosis is drooping off. Drooping off means like a leaf shed off their leaf, like a tree. And a tree, when it shed off its leaves, similarly, the cell, what will do? Cell will shed off that cell which is not required by the body. Okay, that's why the term apoptosis has been derived for it. So, it is a programmed cell death, and if you will properly define the apoptosis, then what is apoptosis? It is a pathway of cell death. Okay, it is a pathway of cell death in which what happened? Cells' own enzymes degraded its own nuclear DNA, nuclear and cytoplasmic proteins. Means when the enzymes of cells itself degraded cells' own DNA, own proteins, then this process is known as apoptosis. Clear? Now comes on the types of apoptosis. There are two types of apoptosis. Whether it's a physiological or it's a pathological. Okay. So it's of two types. Whether it's a physiological or it's a pathological. Okay. So physiological, when physiological apoptosis, example, like what will happen? Like monthly shedding of uterine cells during menstruation. Okay, during menstruation. So this is example of your physiological, right? Like during menstruation, what will happen? There is a shedding of the uterine cells. So this is comes under which category? Physiological. Or second example you can give, like during embryogenesis. What will happen? Like during embryogenesis, what will happen? This is your like development of your fingers. Like when hand will develop, then in between fingers there is a web is there. Okay, when in embryo the web is there. So by the process of this apoptosis, this web will dissolve. Okay, this web will dissolve and normal hand will form during embryogenesis. So the dissolving of this web is also an example of your physiological example. Okay, so these are the two examples of your physiological apoptosis. Now pathological, when pathological apoptosis will occur? When there is a DNA damage. If DNA is damaged, inside the cell by any reason, any radiation or any agent was there, then in that case, if DNA is damaged and it is not properly repaired, then apoptosis of that cell will occur. Okay. Second, if there is a accumulation of misfolded protein. If there is an accumulation of misfolded protein. So if protein is misfolded, then what will happen? If protein is misfolded, then if it will remain inside the cell, then it will cause a number of disorders, isn't it? So cell will dispose of such kind of cell. If then in that cell there is a misfolded protein. So what is the example of this pathological apoptosis? The example of this pathological apoptosis is when there is a DNA damage due to any cause, or there is a accumulation of misfolded protein. Clear? So, two types of apoptosis, physiological and pathological. Now, what are the stages of apoptosis? Now then, in which stage this apoptosis will occur? So, what are the stages? So, what happens? Suppose this is a normal cell. Okay, this is what? This is your normal cell. And when apoptosis starts, when suppose signal comes, there is a lack of survival signal. There is lack of survival signal comes. That means कि now cell has to destroy from the body. Okay, if that signal will come, then what happened first? First, what happens? Cell will shrink. Okay. The first thing what happens? Cell will shrink. Means cell reduce its size. Cell will shrink. Second thing what happens? There is a chromatin condensation. Chromatin condensation. Okay. So this is the first stage. When a healthy cell receives any lack of survival signal, that means that cell have to undergo the process of apoptosis. Then first thing what will happen? The cell will shrink, cell will reduce its size, and there is a chromatin condensation. Okay. What is the next step? Next what will happen? The second thing, there is a blebbing of this plasma membrane. Okay. So what second thing happens? Second stage will membrane blebbing. Okay. Membrane, this plasma membrane will undergo blebbing, and there is a organelles disintegration. So what will happen when there is a healthy cell and that healthy cell receive a signal? That signal cause now this cell has to undergo apoptosis. Then first thing what will happen? Cell will shrink, that is it will reduce its size, and there is a chromatin condensation. Second stage, there is a plasma membrane. The plasma membrane undergo blebbing. So there is a blebbing of this plasma membrane, and the organelles which are present inside the cell undergo disintegration. Okay. So these are organs and these undergo degradation. Okay. Alright. Next step. Then there is a formation of, in third step, there is a formation of apoptotic bodies. Okay. Like this, small apoptotic bodies will be there. Okay. And finally, phagocytosis will occur. Phagocytosis. Who will phagocytize? By macrophages. Macrophages. Clear? So, I'll just repeat it once again. What are the stages of cell cycle? Suppose this is the normal healthy cell. Okay. If this normal healthy cell receives the signal, that is, its signal comes that now this cell does not have to survive. Okay. So, this cell undergo the process of apoptosis. Then first thing what will happen? First, cell will shrink. That means cell will reduce its size and there is a chromatin condensation. Next stage, there is a membrane blebbing. That is the plasma membrane blebbing will be there. And all organelles start disintegrated by the enzymes which are present inside the cell. So, organelles will disintegrated. Finally, what happened after this? There is a formation of small apoptotic bodies are there. Okay. These are apoptotic bodies are formed. And what happened with this apoptotic bodies? Does it need it in the body? No, it should be shed off from the body. No? Then what will happen? Then these apoptotic bodies should be phagocytized by the macrophages. Okay? So this is the stages of your apoptosis. Now comes how the mechanism of apoptosis will occur. How this whole process of apoptosis will occur inside the cell. Okay? So mechanism of apoptosis is somehow complex, but it is highly regulated process. It's a complex. How the mechanism? What is the mechanism? Mechanism of apoptosis. So what is the mechanism? You will write mechanism is complex, but tightly regulated. Okay. Tightly regulated means it's a complex process, but it is regulated properly. Okay, body has a system, body has a mechanism so that this process of apoptosis is regulated properly. All right. So there are what happened? There are in apoptosis two type of mechanism will be there. One is extrinsic pathway. One is extrinsic pathway and another is intrinsic pathway. Intrinsic pathway. Okay, so one is extrinsic pathway, another is intrinsic pathway. By these two pathways, apoptosis will occur inside the body. And this intrinsic pathway is also known as mitochondrial pathway. Okay, so MCQ can be asked, which of the following apoptosis is also known as mitochondrial pathway? Extrinsic, intrinsic, none of the above, both of the above. So option is intrinsic pathway. Okay, so intrinsic pathway is also known as mitochondrial pathway. Okay, and after that, what will happen? Both these pathways are regulated by some proteins that are known as caspases. Okay, caspases. Okay, caspases. What are these caspases? These caspases are cysteine, aspartate, proteases. Okay, proteases. So what are these caspases? Caspases stands for cysteine, A is for aspartate, P for proteases. So the caspases are nothing but these are cysteine, aspartate, proteases. Clear? What these caspases will do? These will split. What they will do? They will split the peptide bond. They will split the peptide bond at the C terminal end. You know, if there are C terminal and terminal 2 terminals are there. So these will split the peptide bond at C terminal end of aspartate. Okay. These will split the C terminal end of aspartate, and what it will result? It results in the formation of various. When this is split, then there is a formation of various procaspases and caspases. Okay, and that play role in apoptosis. Clear, everyone? So what happened in the mechanism of apoptosis? You will write apoptosis is a complex process, but it is tightly regulated. Okay, regulation is properly happened inside the body. So there are two pathways, whether extrinsic pathway or intrinsic pathway, and intrinsic pathway is also known as mitochondrial pathway. Okay, so whether the cell undergo extrinsic pathway or intrinsic pathway, in both these pathways there is a role of these caspases. What is caspases? These are cysteine, aspartate, proteases. Okay, and these are what they will do? They will split the peptide bond at the aspartate protein. They will split the peptide bond at the C terminal of aspartate and results in the formation of various procaspases and caspases, and then these procaspases and caspases further causes the cascade of further apoptosis. Clear? Okay, now comes how this extrinsic pathway will occur. First, we will read the extrinsic pathway. So, what happened in extrinsic pathway? Yes. Okay. So, extrinsic pathway. Listen, example, when this extrinsic pathway occur, like when when there is a activated self T lymphocytes. Okay. Then when the T lymphocytes cell's own T lymphocytes will be activated, then these activated self T lymphocytes undergo degradation by this extrinsic pathway. Okay. If these self-activated T lymphocytes will not degraded by apoptosis, then what will happen? There is a development of autoimmunity. Autoimmune disorders will develop. Okay, we will read it thoroughly in the immunology series. Okay, or second thing, by the cytotoxic T cells target killing. Okay, so when the cytotoxic T cells, T lymphocytes, what will do? When the cytotoxic T cells causes target killing, then the target killing of that cell by the cytotoxic T cell happen by this extrinsic pathway. Okay, so these are the two examples in the body where the extrinsic pathway of this apoptosis will occur. Okay, now we will understand how this process of extrinsic mechanism will occur inside the body. Okay, suppose this is your cell, this is your plasma membrane. Okay, this is your plasma membrane, and this is your activated. This is what? This is your activated T lymphocytes. Okay, this is your T lymphocytes, and body has to undergo started killing of this activated lymphocytes. So what will happen? These T lymphocyte will present some kind of death signal. Okay, it will present this kind of death signal. Okay, these T lymphocytes, these are the self-activated T lymphocytes, will carry the death signal, and that death signal in the form of fast ligand. What kind of this death signal? These are FAS, fast ligand. Okay. Being a ligand, when this activated self-activated T lymphocytes circulating inside the body, then these signals are detected and attached with the death receptor. Death receptor. Okay. So when the activated T lymphocytes are there, and these carry what? These carry this death signal. When it is circulated inside the cell, then what will happen? The cell will detect it. Oh, it's carry death signal. That means it has to be destroyed by the process of apoptosis, because it is not useful for us, no? Because activated, otherwise it will cause autoimmune disorder. So these are detected by this signal. They carry. Suppose this is the activated T lymphocytes in carry such kind of signal, and it is rotating inside the cell. It's roaming here and there, and then cell will detect, oh, this carry this kind of death signal, that is fast ligand is detected. Then cell will detect it, and death receptor will come, and then this death signal will combine with this death receptor. Okay, when this will combine with it, then what will happen? Then your procaspase, it will act on procaspase 8 and 10. When this death receptor death signal will combine, then it will attack on your procaspases 8 and 10. And when this attack, then this procaspase 8 and 10 will convert into your activated caspase 8 and 10. Okay. So these are in procaspase, these are in the inactive form. When the signal comes and it is attached with the death receptor, then when there is this death signal, death receptor complex will form. What it will do? It will stimulate the procaspases. And then this procaspases 8 and 10, which were in inactive state, okay, now comes on the active state of caspase 8 and 10. Clear? Now what this caspase 8 and 10 will do? It will activate another procaspase, that is procaspase 3. Once it is activated, then what it will do? It will activate another procaspase. And now this procaspase 3 in its inactive form, and this 8 and 10 will convert into its active form, that is caspase 3. And this caspase 3, what will do? It acts on the apoptotic substrate and causes apoptosis. Clear, everyone? So, once again I am telling you what happened in the extrinsic mechanism. Okay, suppose this is your activated, self-activated T lymphocytes. So, it is not useful for the body. Now, body has to destroy it by the process of apoptosis. Then what will happen? These, now how body will detect which lymphocyte we have to degrade it. Other also normal T lymphocytes are circulating, okay? Then how body will detect? Now these self-activated T lymphocytes carry the death signal, and death signal is what? In the form of fast ligand. Okay, so when it carry the signal, when these are roaming here and then cell detected it through this signal, and these combined with this death receptor. So death signal, death receptor complex will form, and when it is formed, what it will do? It will going inside the cell and it will activate procaspase 8 and 10, which are in the inactive form, to the activated caspase 8 and 10. Clear? Next, what this activated caspase 8 and 10 will do? It will cause activation of another procaspase, that is procaspase 3. And now this procaspase 3, it is also in inactive form. Now it is activated as a caspase 3. And now finally this caspase 3 will do cell apoptosis. It will act on the apoptotic substrate, it will act on this, and it will finally apoptosis will occur. Clear? So this is the extrinsic mechanism. Now how the intrinsic mechanism or mitochondrial pathway will occur? What happened in the intrinsic mechanism? So intrinsic. Intrinsic is also known as mitochondrial. Intrinsic pathway, or you can also write your mitochondrial pathway. Okay. So when this, in which case this intrinsic mechanism will occur? When there is a DNA damage. As you have seen my previous video of P53 gene, in that video I have just teach that when DNA damage will occur and that damaged DNA will not repair by the P53 gene, then that cell has undergo apoptosis, and that apoptosis process is through this mitochondrial pathway. Okay, so DNA damage or there is a misfolded protein. Okay. For both these mechanisms, cell undergo intrinsic or mitochondrial pathway. Okay, so what happened in this pathway? In this pathway, there is some gene family that is known as BCL2 gene family. These family genes will play an important role. What this gene family will do? This will control. What this gene family will do? This gene family control the mitochondrial permeability. Mitochondrial permeability. That means these genes will control the permeability of mitochondria. Okay. And in this BCL2 gene family, there are two kind of genes. One are the pro-apoptotic. One are pro-apoptotic gene and another are anti-apoptotic gene. Anti-apoptotic gene. Okay. So anti means these genes will not favor the process of apoptosis. Anti-apoptotic. Anti means they will not favor. Okay. Pro-apoptotic genes, these genes will favor the process of apoptosis, isn't it? So pro means these genes will favor the process of apoptosis, and anti-apoptotic, anti means it will not favor. Anti, these are anti. So example is that of like BCL, BXL. These are the anti-apoptotic genes. And what are the example of pro-apoptotic? Pro-apoptotic is your BAX, BAD, BAC, BID, etc. Okay. These are the BAX, BAD, BAC, BID. These genes are the your pro-apoptotic. So what does it mean? When these anti-apoptotic genes present in cell, then these genes will not initiate this process. Okay, because these are anti, so they will not initiate this process of apoptosis. But when these genes will activated or stimulated, then these genes will cause the process of your intrinsic apoptosis. Okay. Now, how this will happen? Suppose if DNA damage occurs by any radiation, there is a UV radiation inside the body and DNA is damaged. Okay, and that DNA which is damaged is not repairable by the p53 gene. Then what cell will do? If DNA is damaged by the UV radiation, okay, if DNA is damaged, then p53 gene will come and it will try to repair that damaged DNA. But either repair successful or unsuccessful. If repair is unsuccessful, then that damaged DNA, which cell carried damaged DNA, it has to undergo the process of apoptosis through this mechanism. Okay. Then what will happen? Suppose this is the mitochondria. Okay. This is your crystal. And this is your mitochondria. Okay. So if that cell which has to targeted to undergo the apoptosis, that cell will activate the they will activate the pro-apoptotic gene. Okay, like your BAX, BAD, BAC. Okay, that cell will activate what? That cell will activate the pro-apoptotic genes, and when these genes will accumulate, they will target and goes to the mitochondria. And what they will do in mitochondria? They will increase the permeability. What they will do? They will increase the permeability of mitochondria. Got it? What this gene will do? Okay. What happened? Suppose a cell which damaged, having a DNA damage, and that damaged DNA is not repaired, then that cell has to undergo the process of apoptosis. What that cell will do? That cell will accumulate and start stimulating this pro-apoptotic gene, like your BAX, BAD, BAC, etc. Okay. Then what is the function of these genes? These genes will stimulate the mitochondria, and these genes will increase the permeability of mitochondria. Okay. So what will happen? When this permeability will increase, then there is a There is a leakage of cytochrome C. Okay, you know cytochrome C is present inside the mitochondria. Okay, so when the permeability of mitochondria will increase, then what happens? From mitochondria there will be leakage of this cytochrome C will start. Okay, when this leakage cytochrome C will leakage start, what it will do? It will act on your procaspase 9. Okay, and it will convert your procaspase 9 into caspase 9. Clear? So as soon as the permeability of mitochondria increase, there is a leaking. As permeability increase, so membrane permeability will increase, and there is a leakage of this cytochrome C, which is present inside the mitochondria, start leak out inside the cell. When it is leakage inside the cell, then what will it will do? It will activate this procaspase 9, which is in the inactivated state. Now this procaspase 9 is activated into caspase 9. What this caspase 9 will do? Then this caspase 9 will activate the procaspase 3 into activated caspase 3. And this caspase 3 again, similar in the extrinsic pathway, will act on the apoptotic substrate, okay, and will convert this into apoptotic bodies, and finally apoptosis will occur. Got it, everybody? So this is the mechanism of intrinsic pathway. Once again, you have to understand what happened. So what happened in intrinsic mechanism? First thing you have to know, intrinsic pathway is also known as mitochondrial pathway. Why? Because this intrinsic pathway is occur in the mitochondria. Okay, that's why it is also known as mitochondrial pathway. So what happened in intrinsic pathway? There is a super gene family, that is of BCL gene family. What these gene family have? These BCL gene family have two kinds of genes. pro-apoptotic genes also, anti-apoptotic genes also. Anti-apoptotic genes example is that of BCL and BCL-XL. Okay, as these are anti-apoptotic, so will it favor the process of apoptosis? No, they will not favor the apoptosis because these are anti-apoptotic. So when cell doesn't go under any apoptosis, then in that case, these anti-apoptotic genes, that is BCL, BCL-XL will accumulate and there will be no apoptosis. But when cell undergo apoptosis, then what will happen? Then there is an activation of that pro-apoptotic genes like BAX, BAD, BAD, PUMA, etc. Okay. So suppose now cell undergo this intrinsic mechanism and cell carry the damaged DNA with itself, and that is not repairable. That damaged DNA is not repairable. Then what will that cell will do? That cell will increase the synthesis of this pro-apoptotic genes, and these pro-apoptotic gene will go and stimulate the mitochondria. And when these genes go in mitochondria, what they will do? They will increase the permeability of this mitochondrial membrane. When the permeability of this mitochondrial membrane will increase, then what will happen? Leakage of cytochrome C. Okay. So when there is a leakage of this cytochrome C will occur, then what this cytochrome C will do? It will activate this procaspase 9. This procaspase 9 is active state or inactive state? Inactive, na? Then this cytochrome C will activate this procaspase 9 into caspase 9. Once this caspase 9 is activated, now what this caspase 9 will do? It will activate the procaspase 3, which is in inactive state, to the activated form of caspase 3. And finally, this caspase 3 will act on the apoptotic substrate and it will cause apoptosis. Clear? So this is all about your apoptosis, and you have to read everything and you have to write everything in your short note. And it is one of the most important topic for the PG and UG, comes as a short note. Okay. So thank you so much.